Okay, let's start our presentation. Here I'm showing you a typical microscope, a light microscope, which most of the labs are equipped with this type of microscope. This is a basic microscope, and I'm quite sure you have seen this one in your lab or in your any experimental rooms. But I prefer to briefly explain some optical and mechanical component of this microscope. So we have this stage and a stage clip which are holding the sample. Samples usually are cultured in a slide or some other culture plates. We have nose piece, we have ocular lenses, or sometimes it's called eyepiece to be able to see the sample. We have objective lens, which someone can play with this objective lens and provide different magnification. Usually there is 10x, 20x, 40x, and 63x, which uses oil also. We have diaphragm and condenser and a light source, and also there is a intensity control to play with the light intensity. So this light microscope has bright field microscope and dark field microscope. So in bright field, we illuminate the sample. There is a condenser lens. We illuminate the sample or a specimen, and then with the objective lens, we can provide a magnified of the sample. In dark field, we use aperture to control the input light, and then the scattered light will be seen, will pass through the MO, and then will be observed on the eyepiece. So the problem is that when we use biological samples or cells, sometimes they are transparent or semi-transparent. So it's difficult to completely observe the sample. Uh, and an alternative way is that to use a phase microscope. A phase microscope uses some optical component like phase ring and phase annulus to to convert the light and phase changes to intensity. Uh, uh, the common property of this microscope is that the results are not quantitative. The results are just pure intensity, which can be changed by changing the intensity of light. Okay, so let's go into the another type of microscope. We have fluorescence microscope, which uses a light source. Sometimes we use LED source, like LEDs with different colors. They have excitation filter to block uh, unwanted light. We have this sample, we have objective, we have dichroic mirror, and we have emission filters. So these filters are responsible of blocking some uh, unwanted light. So usually what we do in the fluorescence microscope, we have to stain the sample with this dye. So what happens when we add dye? The dye will bind to some protein from the sample, for example, to the mitochondria or nucleus or some other part of the cell. And then when we illuminate this one with the proper light, then the protein starts releasing photons to the detector, and uh, it gives us the result. Uh, there are a few things which we have to consider with this fluorescence microscope. First of all, is that this is one invasive method, so the sample should be stained with the color. And secondly, there is a this concept called photobleaching. In photobleaching, the sample gradually loses the ability to release photon to the detector, and when the time goes by, and if we want to record time-lapse images, we gradually lose this contrast and the intensity on the detector. So let's talk about why there are challenges with different microscope modules. The first of all is that the development of the microscope allows scientists to make new insight into the body and disease. So, by inventing new microscopes, we are able to see some some part of the cells or sample, and also we can observe the disease. That's why there is always challenges with the different microscope modules. Also, it's relatively the resource of designing a microscope is relatively expensive, and it takes so much time to explain and teach to a user, and also it needs consumable supplies. And number three, there are some resistance against using microscope. I have seen some biologists, they say we don't trust the microscope because of this, because of that, and that's why there should be always a challenge for accepting a microscope. Because, like I said, some biologists they don't believe in the result. They say there is aberration, impurities in lens, and this can cause a falsified reading on the microscope. Also, with improvements in the lens manufacture, like more pure glass, we can get rid of the color distribution, color distortion, and poor image resolution. Mirrors also can be helpful in this case. There is image blurring because of spherical aberration and color separation because of chromatic aberration. And also in the 20th century, there are electron microscope which increase the magnification and offer new insight into the body and disease studying. Okay, here I want to introduce you a concept, but I am going to explain a little bit some background. So in conventional photography, we illuminate our object or sample, and an object wave will propagate, which construct the light field. The light field will go into the lens and will be magnified, and the image will be uh, available on the image plane, which could be a camera or could be an eyepiece which someone can observe. But there is a second technique which we call it holographic imaging. Holographic is a concept which means this you can observe a 3D representation of the sample on a two-dimensional photosensitive film or just a camera. If you use a camera, we call it digital holography, and in the conventional way, we just see it holography. So in holography, the laser light, which is a coherent or semi-coherent, is divided into a object beam and reference wave. This is usually achieved by a beam splitter. Object will object object will be illuminated by the part of the laser, and the object wave will propagate, and the reference wave will come at the same time. They reach to each other, and then they provide interference pattern. And then this interference pattern will be recorded by a photosensitive chemical. To be able to observe this sample, the photosensitive will be also illuminated with the same laser light, and the diffracted light will goes into the lens, and the image will be provided, and we can also see the virtual image. There is this digital holographic microscopy, which uses same concept, but we use this technique to study microscopic object. Here you see a general scheme of this microscope. The laser light. Here is a red light. We'll divide it into two arms with a beam splitter, and this passes through the sample through microscope objective, tube lens, and then there's a mirror here which can provide us interference pattern on a hologram plane. So this is the microscope shown in below is uh, one commercial device. As you can see here, there is no eyepiece to observe the sample because this is a digital hologram, and the digital hologram will be represented on a LCD, not on a eyepiece. So on the right side, you see a red blood cell hologram, and then below you see a skin cancer cell.